Thank you so much. And thank you so much for the invitation. I'm happy to share this work with you today. So this is recent work that started from a project that Rebecca de Jesus Crespo, who's a biologist at Louisiana State University, had kind of came to me with, and we started collaborating, thinking about how hurricanes affect dengue transmission. So I had done some previous work on how hurricanes affect West Nile virus transmission. And so she was interested in looking at dengue transmission, which is spread by different mosquitoes than West Nile. And so one project that we're currently working on is how hurricanes affect vegetation and how that change in vegetation affects temperature and the resources available to mosquitoes, how that alters the distributions of mosquito species that we see in certain areas of the United States, and then the resulting impact of that on dengue transmission. So I started working with a group of our youth students, Emma, Lauren, Pat, and Reagan, the summer before last, trying to kind of get a start on the modeling of competition between Aedes aegypti and Aedes albopictus, and incorporating the effects of temperature and resource quality to eventually go on and connect to this larger idea of hurricanes. All right, so I'm gonna first give you some background on these two mosquitoes and how temperature affects competition, how resources affect competition, and then we'll talk about the competition model, just on population dynamics of the species, and then extend it to a transmission model. And I also want to mention Michael Roberts also been a collaborator in this work, kind of picked up and we started working on this after the REU, and then we've subsequently expanded the model to be to a stochastic model where we can look at how the type of individual introducing the disease affects the probability of an outbreak versus disease extinction. Um, I think I have to click. Okay. So, Aedes aegypti and Aedes albopictus are two mosquito species that share habitat requirements. They both lay eggs in artificial containers like discarded tires, cemetery vases, flower pots, right? Anything that holds water. And albopictus is a more species that's more recently been introduced into the United States, I believe around the 1980s, uh, through tires, and has since effectively displaced aegypti in many areas. And there are still many areas, especially in the southeastern United States. And in the southeastern US, there are still some places where the species coexist, such as New Orleans and Miami, and the Dallas area. But in these cities where you see coexistence, there's significant spatial heterogeneity, with Aedes aegypti tending to be found in warmer, more urban areas, and albopictus tending to be found in cooler, more vegetated areas. And albopictus is typically considered to be the superior competitor. They compete at the larval stage, and albopictus can better exploit these resources that are available to mosquitoes. But the caveat to that is that the competitive effect, right, this degree of competitive superiority depends on the resources that are present, and specifically the quality of those resources. And that's typically related to like how rapidly decaying is the resource. So, uh, insects and grass would be considered high quality resource, and algae and a lot of leaf litter would be considered low quality resources. So the competitive effect, both intra and intercompetitive effects between these species will depend on the resources. Okay, and that's a key component of this work. And so while albopictus is the superior competitor under many circumstances, Aegypti has a higher tolerance to heat. It can survive at higher temperatures, right? And also has resistance to egg desiccation, though humidity is not something we've incorporated yet. And so it's thought that urban heat islands might help support the persistence of aegypti in cities, right, preventing albopictus from completely displacing them. And so vegetation has an effect on both temperature and resources available to mosquito larvae. So this figure shows this urban heat island effect and how when you have these areas with a lot of building, a lot of concrete, impervious surfaces, you have this retention of heat. It's very hot compared to areas that might be very close spatially, but have more vegetation, right? In these suburban or rural areas, even parks in urban areas can have lower temperatures, right, due to the vegetation. And then when you think about the habitat for mosquitoes, especially like stored tires, it can be even hotter inside those tires, right? It can be like 3 degrees hotter than the surrounding air. So these urban heat islands can be significantly hotter than areas that are not very far spatially, right, but have a different composition of vegetation. Right, so vegetation provides this cooling effect, and it also affects the food sources that are available to larvae, what is in the containers for them to eat. So if there's no vegetation around, maybe there's nothing really in the containers, and there maybe just some algae growing in the water, or maybe there's some grass clippings if there's no trees, but mowers, especially in cemeteries, can get grass into the cemetery vases. And if it's a closed canopy area, maybe that canopy drops leaf litter into the containers. Maybe it also drops insects into the containers, right? So you can have high or low quality food sources in vegetated and non-vegetated areas. So it's not really a matter of, well, more or less vegetated always goes with higher or lower quality resource, right? It's a little more complicated than that. And so what happens is that within cities, and New Orleans is going to be the primary focus of our work, you have this small scale heterogeneity and these microenvironments that differ in temperature, right? You can think of temperatures as heat index, right? And within the city, are you going to be kind of hotter than the average temperature for that city if you're in an urban heat island, or maybe you're in a cool suburban area? And so if you just look up the temperature for New Orleans, right, not every area in New Orleans is going to be that temperature. There's going to be some areas that are warmer and some are cooler, right? And so these microenvironments will vary in temperature. They'll vary in the amount of vegetation, which is associated with temperature, but also can affect resources. The uh, socioeconomic status of these different neighborhoods can affect the abundance of mosquito habitat. Right? Poorer areas might be associated with larger amounts of mosquito habitat, like higher piles of tires. This graphic is from Dr. Jesus Crespo's recent paper on vehicle tires and their association with mosquito vector abundance. And the top graph shows in the different colors the income of different areas of New Orleans. And then the circles are the counts for discarded tires. Okay, and so you can see an association there. And then the heat severity index is shown on the bottom graph. So you can see there's quite significant heterogeneity in both of these factors. There's also heterogeneity in human density throughout the city, which is going to be important, not necessarily for, well, it might be associated with mosquito habitat. It's also going to be important for the transmission model, right? So not as important for the population dynamic model, but for the transmission model, the vector host ratio is very important. The presence of non-human hosts can also vary, right? Are there other hosts for these mosquitoes to bite? And this is something that aegypti and albopictus actually differ in. They're kind of tendencies to bite non-human hosts, and this feeds into the risk of vector-borne disease. So both aegypti and albopictus are vectors of many diseases. A few of them are dengue, chikungunya, and Zika virus. We're going to focus on dengue, but this model could easily be adapted to a different disease. We just use parameters for dengue. Aegypti is typically thought to be a more competent vector of dengue for a few reasons. So, well, albopictus, remember, is the better competitor. Aegypti is the more competent vector in that it is a better transmitter of disease. It has higher infectivity rates. So, if Aegypti is infected with the disease, it's more likely to become infectious and able to infect humans. And it also has a higher biting rate on humans, or higher percentage of bites on humans. Aegypti pretty much mainly bites humans, but albopictus is more opportunistic and it will bite non-human hosts if available. So the presence of these alternative hosts really can impact the percentage of bites that are on humans versus not on humans. But for albopictus, right? The other complicating factor is that Aegypti has been known to take multiple bites per gonotrophic cycle. So the gonotrophic cycle is referring to kind of the period of time in between blood meals. So when the mosquito takes a blood meal, it then and digests the blood and lays the egg, and then there's this period of time before it takes another blood meal. And so if it's only taking one bite or one blood meal per gonotrophic cycle, then that's the only potentially disease transmitting bite. And so you can think of the biting frequency as just the inverse of that length of the gonotrophic cycle. But if it's taking multiple bites, each one, right, maybe it has an unsuccessful blood meal, um, and it has to take more than one, and each of those is potentially disease transmitting. Okay, so here are the questions that I'm going to try to answer um, throughout the talk today. The first one is, how do temperature and larval resource quality affect the outcome of competition between these two species? Once we answer that, we'll look at how the resulting spatial distributions of these two mosquitoes then impact the risk of vector-borne disease, focusing on dengue. Those two questions are published in a single paper in the Bulletin of Mathematical Biology. I have the reference at the end. And then the third question is a modification of the model that's used for Ames 1 and 2, a stochastic extension where we look at how disease risk changes with how the disease is introduced into the population. So it's typically introduced through human travelers returning from areas abroad where dengue is endemic, right? It's not endemic in the United States. And so if somebody brings it back, we want to know what is the probability that we see local transmission, right? Secondary cases. But it's also possible that a mosquito could become infected, and while mosquitoes don't move that far, since the spatial scale on which we see this variation, right, these different microclimates, it is possible that a mosquito could carry the disease to a neighboring area. Okay, so this is the work from the REU. Developed a stage-structured vector competition model. Species 1 is aegypti. I'm going to color code a few of the figures throughout, and aegypti will always be in red and albopictus will be in blue. Aegypti will always be species 1 and albopictus will be species 2. So we used a juvenile adult model because in the juvenile stage is incorporating, right, the egg, larval, and pupal stages. And the reason for this is because competition is occurring at the larval stage or the juvenile stage, but the adults are the ones that are biting humans and transmitting disease, and that's what we're going to want to couple with the transmission model. So, so we've kind of said we need a stage structured model, right? But we don't really need to break down the juvenile stage any further. So the fecundities here is F1 and F2. The fecundities are the eggs laid that survive the juvenile stage. So we don't have any mortality at the juvenile stage incorporated explicitly. It's all in this fecundity term. Okay. The D's are the juvenile development rates. So 1 over D1 or 1 over D2 would be the expected length of the juvenile stage. And mu1 and mu2 are the adult mortality rates. So we assume constant mortality rate. It's or not density dependent. It is going to vary with temperature. So the parameters that vary with resources are the competition coefficients. So alpha 1,1 is competitive effect of aegypti on aegypti. Alpha 1,2 is the competitive effect of albopictus on aegypti. Alpha 2,2, competitive effect of albopictus on albopictus, and 2,1 is the competitive effect of aegypti on albopictus. Right, so the second subscript is the species that's affecting the first subscript. And the rest of the parameters are assumed to be temperature dependent. All right, so the fecundity, development rate, and mortality all vary with temperature. Okay, so this model has 4 equilibria. The extinction state, where neither species persists, 2 exclusion equilibria, where each species persists in isolation, and a coexistence equilibrium where both species persist. And the stability of these equilibria can be written as dependent on these combination parameters that we call capital M1 and capital M2. Right? So there's a capital M for each species, and it's capital M 1 is equal to 1 minus mu1 over F1, and M2 is 1 minus mu2 over F2. Okay? And in order to determine the stable equilibria for different values of M1 and M2, we compare the ratio M1 over M2 to the ratio of the competition coefficients. Right? So there's 2 possible, so for any set of competition coefficients, if we look at the ratios alpha 1,2 over alpha 2,2 and alpha 1,1 over alpha 2,1, right, then one of them is going to be larger. Right? So the difference between these 2 kind of lines, these 2 axes here, are that the first one has alpha 1,2 over 2,2 smaller, and the second one has alpha 1,2 over alpha 2,2 larger, right? So first you want to look at the alphas and say, well, which situation do we have, right? And then we can think about the value of mu and f, right? These mortality and fecundity rates, right? Figure out what those are, and that will determine M1 and M2, and then look at the ratio of M1 over M2, and you want to compare it to these 2 alpha ratios. If it's less than the minimum of the alpha ratios, right, M1 over M2 is less than both of the alpha ratios, then albopictus will exclude aegypti. That's the stable equilibrium. If it's greater than both of them, then you always have aegypti excluding albopictus. And if you are in this top scenario with alpha 1,1 over 2,1 being larger, then if M1 over M2 falls in between the alpha ratios, we see coexistence. And if we're in the bottom scenario and M1 over M2 falls in between the alpha ratios, we have bistability of these 2 exclusion equilibria, and the outcome of competition will depend on initial conditions. Okay. And so you can think about kind of if one species has a benefit, right, then M1, right, can increase if its fecundity is increased or its mortality is decreased, right? And similarly for M2, so changing the mortality or fecundities of these species, right, will change this ratio of M1 over M2, right? And if we increase it, it favors aegypti, and if we decrease M1 over M2, it favors albopictus. Okay. All right, so now we want to dive into the actual values of these competition coefficients more deeply, right? So here we're saying that one of these ratios is larger, right? But which one is larger, right? Do we know anything about that, right? Can we say anything from the literature about the actual relative magnitudes of these competition coefficients? And there happens to be a meta-analysis of different studies that were conducted on competition between these species that were compiled by Giuliano in 2010. And um, what they found was that the relative magnitude of these different competition coefficients depended on the food quality, right? So that's kind of where this idea for this assumption came from was this paper that for low quality food, which took to be leaf litter, and high quality food taken to be grass and insects, we were able to extract these different assumptions on the competition coefficients, right? So from this paper, really what we got was the relative values that were consistent with their findings. And so for low quality food, we had was that the strongest competition coefficient was the competitive effect of albopictus on aegypti, right? Albopictus is the superior competitor, very strong effect on on um, on aegypti, whereas the effect of aegypti and albopictus was the smallest, right? And then the intraspecific competition coefficients fell somewhere in between. Right? And so if we look at the alpha ratios, we see that alpha 1, 1 over alpha 2, 1 is greater than 1. Alpha 1, 2 over alpha 2, 2 is also greater than 1, right? So they're both greater than 1, but we don't know their relative size, right? We don't know which one's bigger. So we can't say that coexistence is always possible. But for high quality food, it was found to significantly alleviate that competitive superiority of albopictus. And it brought this alpha 1, 2 parameter down to being approximately equal to alpha 2, 1, right? And still, we assumed that alpha 1, 1 and alpha 2, 2 were equal. We just know they're larger. For simplicity, just assume they're equal because we really don't know any more than that from this paper. But what we can see here is now looking at alpha 1, 1 over alpha 2, 1, it's greater than 1, but alpha 1, 2 over alpha 2, 2 is always less than 1, right? So if this first alpha ratio is always less than 1 and the second ratio is always greater than 1, then we know alpha 1, 2 over alpha 2, 2 is always the smaller ratio. And so we are always in that situation where coexistence is possible, right? We still need M1 over M2 to kind of land in between these alpha ratios, but it is possible, right? If we're in between them, we would see coexistence and not just competitive exclusion depending on initial conditions. So what we did in order to run simulations was picked some representative sets of parameter values that satisfy this condition. And for high quality food, since we know alpha 1, 2 over alpha 2, 2 is always going to be smaller, we just picked one set that satisfied these. We picked the alphas so that we had one of the ratios equal to 1/2 and the other was equal to 2. But for low quality food, since it's possible to have either one of these ratios be larger, we considered two different sets of, of coefficients. So one of them ends up with a ratio of, I think, 1/3 and 3. sorry, 3/2 and 3. So they're both greater than 1, right? So we've got 3 and 3/2 and 3 and 3/2. But these alphas are chosen so that we have the same ratios, but there's two different scenarios, one where alpha 1 over 2 is larger and one where it's smaller. And then for the high food quality, we just have a set of parameters, um, just a single one, right? Because we know alpha 1 over 2 is always going to be the larger ratio. Okay, and so this is what we're going to use for numerical simulations. And so the first simulation shown here is one where we are just assuming that the fecundity of each species is 1, right? It's not. We'll look into more realistic value next, but just to show kind of what happens when we consider differential mortality rates, right? Because the first thought that we had, right, the main thing we wanted to incorporate when thinking about this model was the impact of increased temperatures on the differential mortality of the two mosquito species, right? The fact that albopictus was more susceptible to heat, right? It unable to survive at these higher temperatures. So as we increase temperatures, we could think about the mortality of albopictus increasing faster than the mortality of aegypti, right? And so what's shown here is the outcome of competition as a function of the mortality rate of each species. So if the mortality of the two species is equal, then on the top panel here, we have low quality food, and the bottom is high quality food. And the A and B are just referring to can I go back? There we go. A and B are just referring to these two different assumptions here. Right. So for A and B, we have the blue corresponding to albopictus excluding aegypti. The red corresponds to aegypti excluding albopictus. The purple is coexistence, and the yellow is competitive exclusion dependent on initial conditions. So the region of bistability, right? And the dashed lines and the dotted lines are these curves at which exclusion of one species occurs, or where we have this change in stability to right, global stability of these exclusion equilibria. All right. So if for low quality food, if the mortality rates of the two species are equal, right, given that the fecundities are also equal, then M1 and M2 will be equal, right? And we'll be moving along this diagonal and we stay in the blue region, which means if the species are equal for low quality food, albopictus wins. Albopictus excludes aegypti. But for high quality food, if the species are equal, right, these equal mortality rates implies M1 over M2 equals 1, and we're in between those two alpha ratios, we see coexistence, right? And we can also think of temperature increasing the mortality rates of both species, but increasing the mortality rate of albopictus faster, which means we would move through this graph on a line with a slope greater than 1. Right? And so if we are in the high quality food scenario, right, we would eventually move to a region where we had aegypti exclude albopictus. And if we were on the low quality food, right, for either one, we would move either from aegypti excluding or albopictus excluding aegypti to a region of coexistence to eventually a region with aegypti only. And if we have the second scenario, we would move from aegypti excluding albopictus, and then somewhere in between here for that same initial condition, we'd switch to aegypti excluding albopictus. So this just shows how we are moving through those different outcomes of competition, right? Similarly to what was shown on this line here, right? As we move through that on a line through that graph, we can think of M1 over M2 increasing, and we're moving through these outcomes. Okay. All right. So this was more hypothetical, like toy example. We wanted to incorporate actual data for these two species. So incorporating actual temperature dependence into these parameters was a little tricky, right? There's not great data for many of these parameters at higher temperatures, but we did the best we could. And the fecundity rates, the fecundity for albopictus was much higher than the fecundity for aegypti. So for the fecundity here, we were incorporating again the number of eggs that hatched and survived the larval stage and the pupal stage at that temperature. Okay, so there's a few things going into this fecundity rate. So this top panel is showing the fecundity for each species. Again, aegypti is in red, albopictus is in blue. And then the mortality rates are shown in this figure B with the dashed lines. And the mortality rate for albopictus is lower, right? So they have a longer lifespan at intermediate temperatures. And then about 34 degrees, the mortality rate of albopictus shoots up. Then we actually cap it at 1 and assume, all right, it has a minimum lifespan of 1 day, right? So that's why it levels off here. The bottom row of figures is overlaps fecundity and mortality rates and zooms in at low temperatures and high temperatures. Okay, and the point at which the curves of the same color, dotted and dashed curves of the same color cross, tells us the temperature at which each species can persist, either start to persist or stop persisting, right, on its own. Right? So not due to competition, just each species in isolation. When is it going to persist? Right? This is equivalent to the capital M for that species being positive, right? So each species can persist when the fecundity exceeds the mortality rate. Okay, and so that happens earlier for albopictus. So albopictus can persist at colder temperatures. But at higher temperatures, right, albopictus can't persist once these lines cross, which happens at 35.8 degrees. And aegypti can persist for another couple of degrees. Okay, so there is this range of temperatures for which aegypti can persist, but albopictus cannot, right? So not even thinking about competition, it's just too hot for albopictus to persist. Okay. This is taking those mortality and fecundity curves and showing M1 and M2, what they look like, right? Only showing the values for which they're positive, right? So these curves are becoming positive when the dotted and dashed lines are crossing. And then more interesting is the curve for M1 over M2, right? So M1 over M2, right? This is showing how it's changing with temperature, right? So as temperature changes, we move along this M1 over M2 curve, which means we are potentially moving through the different outcomes of competition. And as temperature increases, right, from 16.16, right, which is where this is only shown for when both M1 and M2 are positive, right? We quickly shoot up towards 1, then it levels off around 1, and then at higher temperatures increases rapidly again. This inset is zoomed in at the scale from the vertical scale of 0.96 to 1, because while this looks flat here, there really is a small dip over between 31 and 34 degrees, so it is non-monotonic, right? But so if you think about the high quality food example, the first alpha ratio was 1/2 and the second was 2. So there's little purple stars here shown at 1/2 and 2. So as we increase temperature, right, M1 over M2 quickly quickly exceeds 0.5 and then hovers around 1, right? But for high quality food, if we're near 1, that's the regime for coexistence. And then the temperature has to exceed this value right here, right? this temperature right here, so that M1 over M2 is above 2 in order to have aegypti exclude albopictus, right, in order to move out of that coexistence regime. So if the alpha ratios are not near 1, right, then for most temperatures, right, you're not going to be changing the outcome of competition. Um, for the low quality food example, where both alpha ratios are greater than 1, right, M1 over M2 doesn't exceed 1 until very high temperatures. So there is this very small region of temperatures for low quality food for which you can see coexistence, right? If you are in between the temperature for which M1 over M2 equals 1.5 and the temperature for which M1 over M2 equals 3, right? That's the region of coexistence. It's small, but it's there. And then there's also a small range of temperatures for which aegypti would exclude albopictus because of competition, not just because albopictus can't survive. Okay. So what's shown here is these um, mortality and fecundity curves, and the development rate. I should mention that didn't really talk much about the D, the juvenile development rate. There, that affects primarily the number of adult mosquitoes, right? It'll affect the stage distribution, but it does not affect the stability of the equilibria, right? So when we incorporate these temperature dependent curves into the model, we've got, again, let's focus just on the top row. The bottom is a zoom in. For low quality food, A and B, and high quality food, this is showing the outcome of competition as a function of temperature. So I will also mention that all of this is assuming a constant temperature. It's not assuming that temperature varies throughout the year. So this is not a time series. This isn't what's happening throughout the season. That's future work, right? That's what we're working on now. So far, everything is for set constant temperatures. Okay. So for temperatures, um, for large range of temperatures for low quality food, we see albopictus exclude aegypti, right? And it's not until these higher temperatures that we see aegypti able to persist. Okay. And then if we actually zoom in on this temperature range, right? This is that narrow temperature range, and the black lines are showing those temperatures for which we're hitting those M1 over M2 equals 1.5 and 3. Right? And so we see that, yeah, there is a little bit of coexistence for scenario A, and for scenario B, we have bistability, right, of either aegypti excluding albopictus or albopictus excluding aegypti, depending on our initial conditions, right? But then outside of these temperatures indicated with the black vertical lines, right, A and B are the same outcome. And then for high quality food, we see coexistence for a large range of temperatures, right? They've much closer. So then, and the population sizes for albopictus really aren't any lower, right, than they are for the low quality food scenario when they are actually excluding the other species, right? So our total mosquito abundance is actually much higher here for high quality food. And then there is this region once it gets hot enough that aegypti can exclude albopictus. All right, so some conclusions from aim one are that high quality food promotes coexistence. It can allow coexistence to occur over a range of temperatures where low quality food does not allow for coexistence. Higher temperatures can allow aegypti to persist even when food quality is low. And this mechanism can be either through coexistence or exclusion of albopictus. But if we're just asking, well, when can aegypti persist, then it can happen either with albopictus or on its own. And then we also see that aegypti can persist at these higher temperatures where albopictus is just unable to survive. right? It doesn't matter what the food quality is, albopictus just is unable to survive there on its own because of the mortality and fecundity rates. Right. All right, so probably only going to have time to talk about AIM-2, so I'll probably just not rush it and just kind of introduce three and not spend too much time on it um, for the sake of time. But for AIM-2, right, we now want to know, how do these distributions of aegypti and albopictus impact the risk of dengue? Because you might think that, well, it's aegypti is the more competent species, so whenever aegypti is around, it's going to be higher risk, right? That might be true, right? But what if albopictus is at really high abundances, right? Because it's doesn't have to compete with aegypti, right? So what if you're comparing higher abundances of a less competent species to lower abundances of a more competent species, right? So it's not immediately obvious what the highest risk of disease will be. So we wanted to explore that a little bit. And it actually also depends on these assumptions we're making about the presence of alternative hosts and whether aegypti take multiple bites on humans per gonotrophic cycle. Right? So what we did was coupled that juvenile and adult model to a transmission model. So we've got juveniles and then susceptible, exposed, and infected adults for each species. So again, aegypti is in red and albopictus is in blue, the species 2. And then we have susceptible, exposed, infected, and recovered humans. Okay. Uh, the transmission model is pretty standard. We have a transmission occurring between adults. A1 and A2 are the biting rates, and then these rows are the probability of transmission, susceptibility, and infectivities for the two species. We still have competition occurring at this juvenile stage. And then the humans, both the humans and the vectors have an incubation period, so there's an exposed stage. This incubation period is temperature dependent. So we incorporated temperature dependent in all the vector parameters, but not the human parameters. And so we let the incubation period and the vectors be temperature dependent along, as well as these probabilities of transmission and the biting rates. Okay, so all of those are also temperature dependent. The specific functions are in the paper. A lot of them were taken from a paper by Aaron Mordecai, 2017 paper. They're all in the appendix of the Bulletin of Mathematical Biology publication that's referenced at the end. I don't have graphs of all of them, but we did incorporate temperature dependence in these disease parameters as well as the fecundity and mortality rates. Okay. R naught for this model looks like this. Right? So we've got the square root of transmission through a vector species 1 and then transmission from vector species 2. We can see these vector host ratios come into play here. So N is the total population, NV1 is the total population at equilibrium for aegypti, NH star is the human population at the disease-free equilibrium, and then NV2 star would be the equilibrium population of albopictus. All right, so there's a lot going on in this figure. So this is showing R naught. We focused on just R naught for the transmission model. And so the focus on the top row first, okay, the top row is for what we're calling unscaled biting rates. And that means we're just taking the biting rates to be 1 bite per gonotrophic cycle, all bites on humans, okay? The bottom row is for scaled biting rates. And the scaled biting rate assumption is that aegypti take 2 bites on humans per gonotrophic cycle, and albopictus take half of their bites on humans. Right? Just saying, well, what if it's scaled so that there are some alternative hosts? Albopictus isn't biting humans as frequently, and aegypti are biting humans twice per gonotrophic cycle. How does that impact our conclusions? Okay. So what we see is for just the baseline assumption without scaling anything, um, we've got low quality food on the left and high quality food on the right. Okay. So I only plotted the low quality food assumption for which you have coexistence because in addition to showing the R naught value for the outcome of competition, we're also showing the R naught value that would result if we had either species in isolation. Okay. And so if we don't have competition, then the other low quality food scenario was just going to be one of the exclusion curves. So we didn't learn anything new by including it. So it simplifies things to just have low quality food versus high quality food, right? So first column is low quality food, second is high quality food, first row is unscaled, second row is scaled. All right. And again, the blue curve is R naught for albopictus alone. The red curve is R naught for aegypti alone. And the black curve is the R naught for the outcome of competition vector abundances, right? So again, we're going back to this graph, and then we're focusing on feeding these population dynamics for low quality food A and then high quality food, right? We're taking these vector abundances, using those as the disease-free equilibria in R naught here, right? So feeding those in for NV1 star and NV2 star, and then showing R naught as a function of temperature, where all of these other parameters, except for the human parameters, are temperature dependent. Okay, and so again, this is a bifurcation diagram of R naught as a function of temperature. Okay. So when we have low quality food, we've got primarily aegypti excluding albopictus excluding aegypti for most of these lower to moderate temperatures. And so the outcome of competition R naught is the same as the albopictus only R naught. Okay, so the black is on top of the blue here. And then once it gets hot enough, and we've got this small region of coexistence, right? But then after that, aegypti excludes albopictus, so we hop over to the red curve, right? And so one thing to conclude from this is that a very small difference in temperature can really result in a significant difference in R naught, right? Especially when this food quality is low. Right? And we can have a quick switch from something very low to something very high if we have a change in which species can persist. If we're comparing low and high quality food, well, high quality food is assumed to reduce all competition. And so the overall vector abundances get larger. We'd expect to have a higher R naught. So yes, it is true that at any given temperature, R naught is going to be higher for high quality food than it is for low quality food. But it is also possible that we would have a situation where low quality food at a given temperature could have a higher R naught than high quality food at a different temperature, right? So a low quality food environment experiencing a low temperature, right? So maybe like over here, low quality food at 33 degrees, right, might have an R naught of 2, whereas a high quality food at something slightly higher might have an R naught less than 2, right? So we have to take into account both the temperature and the food quality. And then when we I guess we don't need that. When we consider the scaled biting rates, right, then the kind of the maximum risk shifts. So for low quality food, maximum R naught occurs when we've got albopictus only, and maximum risk occurs for temperatures like 28 degrees. But then when we have scaled biting rates, the maximum risk is going to occur at a higher temperature once we switch to aegypti, excluding albopictus. Okay. And then for scaled biting rates for high quality food, we see not much of a difference between the unscaled, right? We actually see pretty similar values of R naught, right? So it's assuming scaled biting rates is going to have a larger impact on predictions for risk when we have low quality food, and doesn't really matter as much for high quality food. Right here, we actually see a decrease in that maximum value of R naught, right? Because we're assuming that albopictus is going to kind of result in fewer bites on humans, right? So we're kind of discounting the importance of albopictus, and then increasing the number of bites for aegypti. Okay, so I don't have time to talk about this last aim, but kind of what we were interested in. I gave you the overview earlier, looking at, well, does it matter if humans introduce the disease versus a vector? And so we went through and we computed the probability of disease extinction. Right? It's possible that when the disease is introduced, it can die out before leading to a major outbreak, right? If that infected individual who's returning or spreading the disease happens to recover or die prior to transmitting it, right? It's not going to actually lead to an outbreak, even if R naught is greater than 1. And so we kind of took a variation on this model, assuming a constant population size, and we explored more the composition of the population and the percent aegypti versus albopictus, and looked at the how that affected the value of R naught along with these probabilities of extinction. So we used a CTMC model, computed these probabilities. So this is just all the details for that. And then we looked at how it changed with these different parameters. So percentage of each species, overall vector host ratio, temperature, this biting behavior, diving into more the possibility of alternative hosts and the percentage of albopictus bites on humans, and then the type of infected individual. So we get these types of curves, and we can look at how R naught changes with the composition of the population, and see kind of how introduction by humans compares to introduction by these other types of individuals. And kind of one general conclusion that we found, without going into the details, is that introduction by humans that tends to be much more sensitive to vector composition than introduction by any specific vector species. So that's it comes down to the fact that when a human is introducing the disease, right, it's not guaranteed that that human will be bit, but when a vector introduces the disease, that vector is assumed to always bite a human, right? And so, especially when maybe I'll just show you the vector abundance. So the vector abundance, right, we see that as the vector host ratio increases, the black line is the probability of extinction for introduction by humans, and that keeps decreasing with vector host ratio, right? A lower P naught is a lower probability of extinction, higher probability of an outbreak, higher risk, right? And that doesn't saturate with vector host ratio the way that the introduction by either an aegypti or albopictus mosquito would. Right? So kind of some of our next steps are, again, to incorporate this time varying temperature, look at a time series throughout the year rather than just looking at constant temperatures and bifurcations and with temperature. We want to further connect it to the specific neighborhood level data in New Orleans, some of which I showed you. And a larger question that we're looking at is connecting this to questions of how hurricanes affect vegetation and how defoliation affects temperature and resources, and then how that affects vector composition and vector-borne disease risk. And so that's RU was funded by NSF, and here's the reference for this paper. And have a couple of minutes, hopefully, for questions if you have any. So thank you very much.